I would like to now introduce Katie Thomas. She is with the Institute for Child Development. She is a researcher. She's done a lot of work on cognitive development and in implementing some of these trauma strategies into the work that she's been doing. So she's going to talk more about brain development. So we're very happy to have her here.
Well, thank you all for coming today and for joining us in this conversation about the nursery way, interventions, brain development, and how we as individuals who care about children and families can all work together to understand more about the situation and to inform each other about how we can improve the work that we do. As Molly and Michelle mentioned, I'm a researcher. I am not a clinician or an intervention specialist. I work here on campus doing research in a laboratory. We bring families into our laboratory, so it's not a real-world setting. But my goal today is to try to give you a flavor for how the research that has been done looking at brain development from a very basic level of brain development all the way through to these complex interactions and relationships with parents and children, and some work on cognitive and social development in children and how that relates to brain development that can really lead us to have more discussion during our last question and answer period about the interactions between the research world and the applied and intervention world. So I'm going to start at a really basic fact here, which is we all know the brain is amazingly complex. Probably in various settings, you've learned quite a bit about the brain, even from the popular news literature. We know a lot about what's going on in neuroscience these days. We hear about how the frontal lobe is one of the last areas to develop, that it's still developing well into adulthood, that even those students here at the university haven't reached their adult maturation levels of the brain, and that some of us are still going up and some of us are coming back down. And it's a process. It's a process over time. And that's the main thing I want to say here is that we're never going to be able to say, here's this one brain area that does this whole complex thing. If we think about the complexity of relationships between children and parents, between caregivers and children, between teachers and children, we're never going to be able to point to a brain area and say, that's the problem spot, that's the thing we need to fix. It's never going to be that simple. It's just not possible in a complex system like this that has so many different facets to it to think about a simple answer for how our experiences are incorporated into our biology, how our biology affects our next wave of experiences, and so forth. Part of that is an acknowledgement that we do have a biological makeup. All of us in this room differ from one another, not only in our personalities and what we choose to do with our lives, but also in our biology. We talk about genetics and DNA a lot, but sometimes from the medical literature, we get this sense that the DNA is this blueprint, and I think that's a nice metaphor up to a point. But if you've ever had an architect designing something for you for your home, architects don't like to change the blueprints once the blueprints are drawn out, right? We don't want to go there. That was a hard process. And that's something that's not quite the right metaphor for brain development or for DNA. The DNA isn't fixing the possibilities that can happen in our lives and how our brains can develop. There's a certain range, okay? I'm never going to sprout wings, okay? That's in my DNA. But within the range of human behaviors, my DNA does not say that you cannot do this. And for most of us, there's just so much that the DNA cannot code for. We can't code for which of us are going to be good, are going to choose to follow a certain career path, or which of us are going to choose to work with children or not. So there are a lot of things that are dependent on our experience. And our brains have to be able to adapt to changes in our environment, either positive or negative. All of us, even if we've had glowingly positive lives, have had all kinds of changes in our environment, things that might have been unexpected. We moved, we met new people, we were put in a situation where we had an opportunity for something that we didn't ever know. The brain didn't know we were going to have that opportunity. So we really have to have a mechanism in our biology that allows us to adapt to our environment. And I put there both adapting to either positive or negative, because we've talked a lot about trauma today, and it's incredibly important in our thinking of what children are reacting to, what their current state is based on their past experience. But positive things have just as much of an impact on the brain as the negative. Sometimes we have these overwhelming situations that are so unexpected that they stand out to us because they're just horrific, the things that have happened to some of these children. But we forget that there are all kinds of really positive things that happen to children every day, and that's equally important for the brain. One of the hard things, though, is really thinking about how does that experience get in there? What is it that's changing in the brain? How does that happen? And I think we have to go back to some of the really basics about how the brain is built, the cells that are important in the brain and how they interact with one another. So if we think about the basic building blocks of the brain, we have two broad categories of cell types that are building our brain. The neurons, which are those communicating cells that we think of as being so unique to the brain and really being the core of how our experience can get into the brain. But we also have a whole network, a matrix of other cells called glia that are supporting that network of neurons. And it's not unlike social support in the context of a child's life. You have these neurons that are so critical and are undergoing all these aspects of development, and they must have that support structure to support their growth and development. We don't need to go into any of the details about how these cells are unique in the body or what's going on, but if we look at the leftmost figure here, which is a figure that was used in the movie The Secret Life of the Brain, which is also a book, which is what's cited there. But it's a depiction of what it might look like for these neurons to be connected to one another. Of course, all the space in between is filled with other things, right? There's not all this space in your brain between these cells. They're all packed in there very tightly along with their support networks. But what you're seeing is that these neurons are highly interconnected. And the way that experience can get into that brain is predominantly through altering those connections. You're probably familiar with this, but that's just something to keep in mind, that our main point of change is really in thinking about the interconnectedness among different brain regions, among different cells. Another point to keep in mind is that the wiring up of the brain is a developmental process. It isn't something that we're born with, that it's already pre-wired, we pop into the world and everything works beautifully. In fact, it is amazing that the brain is so complex in a young infant. We think about a newborn and how much complexity is already there. It's kind of overwhelming. But the reality is that the continued wiring in our adult brains are really built on this developmental process of change over time, how we alter those connections and how our experiences work their way in is through this connectivity. This figure is just trying to show you some of the waves of change that can occur in the connectedness among regions. It's a process called synaptogenesis, the synapse being the connection between two neurons. If we think about very early in development, we have, in the prenatal months, we have the birth of neurons and the migration of those neurons to their final places in the brain and the beginning of the connectivity. So it's really in about the last trimester of pregnancy that we're seeing this huge increase in connectedness among the neurons. And so that's what's depicted at the beginning of the red bar along the bottom. The first curve that goes up that's labeled sensory motor cortex is just indicating that there is this rapid increase in connections among neurons in the first 2 months, prenatally actually there. We're still talking prenatally. So in the sensory motor regions of the brain, the regions that have to do with our vision, our hearing, our motor movements, our sense of touch, there's this huge increase in connectivity. So when the infant is born, they have lots and lots of connections there. And in fact, you see that we see similar increases and peaks in all different areas of the brain, but they're not all happening simultaneously. That's a really important aspect here. The broadest and latest peak here that's shown is for the prefrontal cortex, and that's just indicating to us that there's a much more protracted development of this connectedness in certain brain regions compared to others, that it's not a fixed time frame when there's no hope of change after that. The other thing to keep in mind is that we show this peak and then we show a drop-off. And we sometimes think, ooh, losing brain connections, that's bad. But actually, in this case, it might be very good. There's a certain process in brain development that involves overproduction of connections. We have all of these connections, but not all of them are necessary or efficient. If we think about a network of phones, right? If we just connect all the phones to each other, it might be really hard to identify. And I dial on my phone, I want it to ring a very specific person. I don't want it to just ring everybody in New Jersey, right? Or anybody in New Jersey. I want it to ring my grandmother. And so there's this process early on where we have overconnectedness, and we need to refine that. And experience is the #1 way that we get to that refinement. It's through our everyday interactions with our world that we start to refine these connections, and we keep the ones that are used the most. There are lots of ways that the normative processes of brain development can be disrupted or can experience unexpected events, right? If we think about the typical pregnancy in the best possible situation, there are a lot of similarities from one to the next. Many of the factors are quite similar, but you can all think of examples of how individual pregnancies are going to differ from one another. Just like individual families differ from one another or homes differ from one another. So we do know that there are a lot of factors, both physical factors like toxins, but also behavioral factors, maternal stress, maternal infection, medical factors that can impact the development of the brain, the organization of neurons, and the connectedness among neurons. So there are already things prenatally that can be impacting the starting state that the brain is in when it arrives in this world, in this external environment. Not all of those are things that are imposed. Here I talk about errors of commission, things that were done that were unexpected, things that happened that occurred. So environmental toxins occurred and the child was exposed to that was unexpected. A maternal infection is not something that's typically expected, and we hope it doesn't happen. It's something that was committed. But we can also have errors of omission. And this might be one of the things that's more prevalent in our thinking about social and emotional development. We already talked a little bit earlier about the social deprivation, even the sensory and cognitive deprivation that can occur in orphanage rearing. And it certainly was very extreme in some of the studies of Romanian orphanages. But it happens in most institutional settings, that there's not the same kind of one-to-one interaction between a caregiver and child, that there isn't always the same opportunity for cognitive stimulation. So prolonged rearing in an institutional setting is usually associated with some degree of social and sensory or cognitive deprivation. We can also think of physical situations that might impact brain development that are involving an omission of something that's important. Hypoxia is a medical condition, meaning lack of oxygen. If a child isn't getting enough oxygen, it's going to affect all of their body's organs, including the brain. And the brain thrives on nutrients like oxygen, like glucose, that are important for brain growth and development. Malnutrition seems like another obvious one that we can think of, but sometimes we don't think about the errors of omission that occur on a much more subtle basis. That subtle rejection in the family, peer situations that might also be errors of omission. When we think about the development of functions in the brain, what's actually going on in the brain? A key point is that we're not pre-wired ahead of time for function. Even things that seem very basic, like vision, being able to open our eyes and see in the world. It turns out that we need input from our environment in order for those aspects of typical brain development to be initiated and to start. I just put this cute picture of a baby here to remind us that even in typical development, normative interaction with the environment is critical for very basic systems to develop. So vision requires light coming into the eye in order for the visual system to develop fully and normally in humans. If we, for whatever reason, are deprived of that necessary stimulation, those typical visual areas of the brain will not develop normally. So we can think about cases that might be really outlandish, meaning they don't happen very often. But what if a child's eyelids didn't develop properly? And so they couldn't open their eyes and get the patterned light coming into their eyes. Their brain would develop differently. Their visual cortex is not preformed at birth. It has to have that activation, that function along those circuits in order to develop vision. I put up this example of a really interesting research study trying to look at this flexibility. How is it that the brain develops even the most basic sensory functions? And it looks very odd because these are brains from ferrets. This is research done by Migranka Suhr, who's at MIT. And what he was really trying to discover is whether or not certain regions of the brain are predestined in a way, preset to become a particular type of cortex, have a particular function. So in this study, he was looking at visual cortex and trying to say, well, is there a reason why the back of the brain becomes visual cortex? And is that reason because that part of the cortex is developed so differently, or is the reason because vision goes to the back of the brain and therefore it develops there? And so what you're seeing here on the left-hand side is the typical ferret brain with a depiction showing that input at the top, which is where the eyes are there, that's we're looking at down from the top of the head. So up by the nose is where the eyes are, and the vision is coming in there, the light is coming into the eyes, and is moving to the back of the brain where the traditional visual cortex is. You can kind of ignore the colored things on the bottom. That's just proving that that cortex actually has the functions that are associated with visual cortex. But what he did was he rewired, very early in development, he rewired the eyes to go to a different part of the brain. And he rewired it to go to the traditionally auditory part of the brain. And what he wanted to know is if that input from the eyes would make that cortex into visual cortex, even though it's not traditionally visual cortex. And the answer was yes. If you can do this early enough in development, there's no preset that that has to be auditory cortex. It can develop all the functions of visual cortex. There are limits. Timing matters. In brain development, timing is one of the key factors that after a certain period of time when the system has had input and it's developed in a particular way, it doesn't just automatically become reused for something else. There's a lot of plasticity in the brain, and certain regions can take over the functions of others over time, but it's not completely open-ended. So anytime that we can intervene earlier, we're much more likely to have a window of plasticity or flexibility in which the brain can adapt. We talk about unexpected events like toxins or maternal infection, but also some environments are unexpected. We can think of child abuse clearly as an unexpected environment. I don't think that our brains evolved to include child abuse as an expected possible environment. Maybe one that seems more clear-cut is the case of prematurity. So a child who's born a month, 2 months early, comes into this world that's an unexpected environment. The extrauterine environment is unexpected for that brain, which is expecting the 40 weeks gestation. And so we can think about whether or not there are aspects of that unexpected environment that could have negative impact on brain development or even positive impact on the brain development. So everything isn't always negative. In this case, there are lots of risky things for a child being born prematurely. There's a lot of brain development that has to happen outside of the uterine environment. That's really unexpected. But on the other hand, this brain is being exposed to different kinds of environmental stimuli at a point in time when it was not expected. That could either be negative, it could be overwhelming to the system that's not organized in such a way as to react to that, or it could be positive, giving this child an advantage that they've already got that system in place at a much earlier stage of development, allowing them a window of opportunity. So we need to think about both opportunities, risk, and possible benefits as well. I wanted to talk about what we know about trauma and its effects on the brain development because that's the focus of our meeting today. We do know quite a bit already about how trauma in children can impact brain development. There are a number of studies now looking at using MRI imaging. MRI is a method for non-invasive imaging. I mean, there's no exposure to radioactive isotopes, there's no injections required or things like that in order to do imaging, so it's a really great method for seeing the inside of the body, the living body. And so in this particular study, they were using MRI scans, not unlike what you would get if you needed an MRI of your knee or something for an injury. So it's looking at just the structure of the brain. And what they did was look at children who had a history of abuse or neglect, those who were also in the same hospital but for other reasons, and children who were typically developing without a history of abuse or neglect from 2 different sites. And so if you look at the far left column in this figure, what you're seeing is the total volume of the corpus callosum. You might remember the corpus callosum is that large fiber bundle in the middle of the brain that's really the main connection between the left and right hemispheres of the brain. Otherwise, our 2 hemispheres are essentially separated. They're doing 2 different things. They're independent. But through this corpus callosum, we get the connections back and forth. And what we see from this study is that the abused and neglected children are showing significantly smaller corpus callosum volume. They're showing less connectivity, less interconnectedness between the left and right hemispheres of the brain than either the typically developing kids or the other hospitalized children who were not there for abuse and neglect. This is true of the whole brain as well, not just the corpus callosum, but the entire brain volume is affected in children who have been abused or neglected, suggesting that there is some way that this postnatal experience is getting into the brain and altering its overall structure. We also know that our experiences drive learning in our environment. That seems pretty clear from our teaching and intervention situations, but that learning and experience is also changing the brain connections. If we think about typical development, there's a couple of really nice examples that show us how it is that our learning in our everyday experience is altering our brain. If you go to the Wonder Years exhibit at the Science Museum, you'll see evidence like this. So one of the fascinating things to me as a cognitive development specialist is that very young infants, 6-month-old infants, are better than adults at certain tasks. And one of the tasks that they're better at is distinguishing among speech sounds in different languages. So if we either use electrophysiology, electrodes on the head like this image is showing, or if we use behavioral methods that have been developed for work with infants, we can show that babies can discriminate among sounds in languages that are not their native language, whereas adults have a really hard time with that. If there are sounds that are important in the language of one language but not in the other, it's really hard for the non-native speaker to hear those because they don't make any difference in their own language. So for example, in Japanese, the distinction between L and R sounds is not meaningful to the language, but in English it certainly is. So it's very difficult for most native Japanese speakers to learn to hear that difference. And it turns out that by 11 months of age, these babies are already specializing. They're already specializing in the language that they're hearing around them, so they no longer discriminate those sounds either. Despite the fact that early on they could hear all those differences and they would discriminate, they become irrelevant. And the idea that we think is behind this is that they become more efficient in the way that they process the information around them. They're learning what's relevant and what's not, and they are no longer attending to and discriminating among things that aren't relevant to their own environment. Another example that I find really interesting is this example on the right that's showing faces. Most of you looking up here will have no difficulty discriminating the 2 men on the top row and saying that those are 2 different people. But you might have more trouble deciding whether the monkey faces on the bottom are the same monkey in slightly different orientations or different monkeys. They have a little bit different coloring around their face, so maybe it's easier to discriminate those. But it turns out that adult humans are incredibly bad at discriminating among monkey faces. And yet we're exceptionally good at discriminating among human faces. So just like in the speech world, young infants, 6-month-old infants, are very good at discriminating both human faces and monkey faces. So they're good at discriminating those monkey faces just like they are at the human faces. But by 9 months of age, they're specializing. They're becoming good, they're even better at discriminating the human faces, and they're losing their ability to discriminate among the monkey faces. We presume because A, they're not seeing very many monkey faces in their environment, so they're not getting that experience, and it's not relevant. It's not relevant to what they need to do on a daily basis to identify their caregivers and so forth, which is a really important aspect of the infant's world at this age. When we think about trauma and how the brain is changed, Michelle mentioned earlier that there's this lovely line of work by Seth Pollack and his colleagues at University of Wisconsin-Madison, really looking at how it is that the brain of an abused or neglected child has been altered by their experience. And through a number of studies, he's shown that physically abused children require much less information in a face in order to recognize an angry facial expression. So if you look up on the faces on the top here, you can see that we've pixelated the center of the face, and you can't really figure out anything much. This is a little bit over-exaggerated in my slide. It's not exactly what the children saw in the study, but they saw something like this, where the pixels were removed little by little, and children were asked, what expression does this face have? What expression does this face have at each stage, until they could identify the emotional expression. So if we look at the final stage on the right, that face is somewhat angry. It may be slightly on the neutral side of angry. Maybe some of you find it more angry than others. There are individual differences in how we perceive these. But what we see on the bottom in these figures is that in the dashed lines, we see that the abused and neglected children, in this case it was physical abuse, the physically abused children can identify those angry faces at an earlier stage with less information in the face. They can correctly identify it. But importantly, they aren't just better at all facial expressions. It's not like they're experts at reading people's emotions. They're experts at reading anger. If we look at sad, the physically abused children are actually much worse at identifying sad faces. It takes them much more information, much longer, to identify the sad faces. So there are these changes that occur in our processing of the world around us that are reflected in the processing in our brain. And in fact, when they used electrophysiology, these are scalp recording electrodes, so they're recording the electrical signal that the neurons are producing as they're firing with a net something like this. And they're measuring the response to different facial expressions in a task where children were identifying particular faces. So when it says angry target, that's a task where the children were identifying the angry face. Happy target means they were identifying the happy face. And what you can see is there's a much greater response between the difference between the two lines here is what's important, and there's a much larger response during the happy targets than for the other two. This is just suggesting that, like the behavior in the brain recording, we can see that this brain is more sensitive to those angry facial expressions. Physical abuse isn't the only kind of situation that children might experience that might involve trauma. Certainly we know a lot about neglect, somewhat less than we know about physical abuse because it's a little bit harder to classify exactly what the experiences are that a child experiences during neglect. Certainly there's a lack of social stimulation, so we might think about the relationship between neglect and the situation of orphanage rearing, where there's also this profound social deprivation. My colleague Megan Gunner and I have been collaborating on a project now for the last several years looking at children who were internationally adopted from orphanage care. So these are children who are living in really wonderful, highly resourced, highly motivated families here in Minnesota whose families are very invested in their positive development, but who experienced some period of early deprivation by living in orphanage settings. And they really varied in the duration of time that they spent in these orphanages. Some of them were adopted quite early, by 6 months of age. Others spent a year, 2 years, 3 years in orphanage care prior to adoption. We are now testing these children in early adolescence, in 12 and 13 years of age. And what I'm showing you here is an example of using MRI data to look at the volume of the brain in different regions of the brain, just like I showed you for the maltreated children in the previous slide. And what you're seeing here is that for our Minnesota control sample, kids from similar SES families around Minnesota, you can see the volume of the prefrontal cortex on the left and the right hemispheres in the white bars. And then we're seeing for post-institutionalized children, that means internationally adopted from orphanage care, early adopted or late adopted. early adopted in our model is less than 12 months of age. So some of these children spent as much as a year in orphanage care prior to coming to their families. And what you're seeing is that the prefrontal cortex, this incredibly important region of the brain that's late developing, that is important for regulation, for planning, for decision making, is showing much smaller volume in the post-institutionalized children than in the controls. And interestingly, in this study, it doesn't seem to matter how much time you spent in that deprived setting. Even 6 months of deprivation is sufficient to show this effect. And it's not the whole brain. The brains tend to be smaller in these children, but if we account for overall size differences, the prefrontal cortex is specifically affected more than other regions of the brain. And that's really critical in thinking about how research can inform our understanding of the effects of early deprivation and stress. We also have talked about connectivity and how important these connections are between different parts of the brain. This is a study from another group in England that looks at Romanian adoptees to England. And what they've shown is that this period of early deprivation in orphanage rearing is associated with decreased connectivity between the frontal and temporal lobes. Well, what's in your temporal lobe? All of your memory systems, your emotion systems, the limbic system is predominantly in your temporal lobe. What's in your frontal lobe? The cognitive control, the regulation, that cold decision-making part. And so we're seeing less connection, less communication between the prefrontal cortex and these limbic systems and the memory systems in the brain. How does that happen? Well, one thing that we can think about are some of the natural processes that are involved in brain development. So at the beginning, I said that there are these waves of connectivity that are then followed by decreases in connectivity, and that decreasing connectivity is incredibly important, that it's efficiency of connections. What I'm showing you here is another kind of imaging called diffusion tensor imaging, or DTI. It's another MRI method, but it looks at the connections, those fibers that are linking between different parts of the brain. And what I'm showing you here is that a bundle of connections that are going from the temporal lobe to the frontal lobe and back again. And you can't tell just from this image, but the quantitative data suggest that some adoptees are showing much more connectivity, like extra connections. And yet I told you that in the previous slide they have less connectivity. And so there are different measures of connectivity. There's the efficiency of the connections, and there's also the number of fibers. And so it seems as though some of these adoptees are showing extraneous fibers that aren't actually efficient in what they're doing, and they're not all going in the same direction. They're kind of all over the place, suggesting that there might have been some change in this pruning process, this trimming back of the unnecessary synapses or connections. One more type of imaging that we use in our studies of international adoption is functional MRI. This is another MRI method that just allows us to actually look at which parts of the brain are the most active when people are engaged in a particular task. So we have children lie in the MRI scanner and perform a cognitive task. And while they're doing that task, we measure which parts of the brain are particularly active. And what we're seeing in this image, in all the blue areas, are differences in brain activity on a hard cognitive task, a cognitive control task. It's not an emotional task, it's a straight cognitive task, but it involves prefrontal cortex. That in all the blue areas, we're seeing greater activation by the group that spent longer in the orphanage, those who experienced more deprivation. And the regions that we're seeing here are prefrontal cortex and parietal cortex, regions that are really involved in cognitive regulation and self-regulation. What we're seeing in the orange are areas where the earlier adopted children are engaging that region more than others. And you may not have enough neuroanatomy to know what you're looking at there, but those orange regions are very, very close to the hippocampus, that memory-related region that's also involved in learning associations. As we're learning new information, building associations between the stimulus that we see and the response that we make. That's a really important learning area of the brain. And so what we think is happening here is that children who experienced longer periods of deprivation require more engagement of these frontal and parietal regions in order to perform at the same level. It's not that they can't do it, but they have to recruit those brain regions even more. So if you imagine a time when they're in a period of stress and our ability to recruit those regions is reduced, those children are going to have even more difficulty engaging those prefrontal processes than the non-adopted children. I want to end on the positive note because I've just shown you that there are lots of ways that the brain is altered by negative experiences. But I said at the beginning that I believe, and I think the data support the fact that positive experiences also lead to positive changes in the brain. A lot of this work is based on animal literature, and there are two types of animal studies that I want to talk about. The first one is pictured here, and it's work by Bill Greenough, who's now an emeritus professor at the University of Illinois, Urbana-Champaign. And he was really a pioneer in thinking about how altering the environment of the organism might change the physical anatomy of the brain, but also the connectivity of the brain. And so what he did was kind of simple. He changed his rat laboratory from one that uses the standard cages, which they look kind of like plastic tubs that are transparent so the rats can see out into the environment, but usually rats are housed with one or maybe two rats in each of these cages, and they spend most of their time in there except when they're doing experiments. But what he did instead was he took a big container, a very large kind of cart that had a recession in it, and he put all kinds of children's toys down in the bottom, and he housed these rats in communities. There were multiple rats, meaning there was social interaction among the rats. There were lots of new toys and environments to explore, and he changed the toys and environments every single day. So every day these rats experienced some new opportunities to explore their environment. And what he found was that difference in complex environment, the ability to have cognitive and sensory stimulation, to have social stimulation, resulted in alterations in the brain. So actually, on each neuron, there were more connections, more opportunities to have connections with other cells in the brain. And he also showed that it alters the vasculature, the blood flow to the brain. It improves the blood flow and connectivity among different regions. And this work has gone on to drive the work looking at exercise and the importance of exercise in altering the brain. You may not realize it, but if you just allow the rats to run on a wheel, which they will do happily, they show much better brain connectivity and healthier brain development. And there are now a number of studies looking at the role of physical activity and exercise in humans in brain development. There are a number of interventions that have looked at brain activity and trying to show that the impact that we can make is not just behavioral, what we can see, but that there's an internal impact, that behavioral change that you see in children when you intervene with them or with families or with the parent-child interaction actually is instantiated somehow in their biology, in their brain. And there are a number of interventions now. The one that has the most data looking at actual brain activity is an intervention on dyslexia. So we know a lot about phonological dyslexia, a particular kind of dyslexia that involves difficulty in hearing the sounds of phonemes. So being able to recognize that this letter combination involves this sound. And if you look at children who are normal reading, typical readers, and children who are dyslexic, who are having difficulty with reading, on the top row there, you see two circles where we're just highlighting that in typical readers, we see activation of these specific brain regions that are associated with that phonological processing. And in dyslexic readers who are struggling with this, we're not seeing activation in those regions. The question is, what happens if you can intervene and provide a curriculum for trying to train phonological awareness in these children? Can you alter the brain systems that are engaged in that? And the answer seems to be absolutely yes. If we look down on the bottom, now we're seeing dyslexic children after their behavioral remediation, their intervention, that was done by all of the folks like you, practitioners out in the field, people who are working one-on-one with children and families. And what we're seeing is that those behavioral changes that children are experiencing in their reading ability are reflected by changes, increases in activity in regions that typical readers normally activate. So we're sort of normalizing, in this case, the activation of the brain. That's not always the case. There are times when intervention doesn't work to normalize the brain, but often it will work to recruit a new area. A new area can come online to help support those functions that aren't working in the existing system. The other type of experience that I wanted to talk about, and research on experience, is a whole line of work looking at maternal caregiving in rats. It sounds really strange to think about what mother rats do with their rat pups, but they actually do a whole lot. So one of the things that mother rats do, you may not remember this if you've never had a pet rat, but rat pups are born, like human infants, entirely dependent on their parent. They aren't mobile on their own. They don't have their eyes open. They're not experiencing the environment. They need their parent to take care of them. And so the maternal rat is spending a lot of time licking and grooming her pups, giving them sensory stimulation. She's providing a nest for them to help with their temperature regulation, so physiological regulation. So all of that information that we were talking about how parents early on are taking on the regulatory responsibilities for the parent-child dyad and trying to help regulate their child and teach them how to incorporate regulation into their everyday lives happens with rats too. And we see that if you do something as simple as taking away half of the bedding material in the rat cage, that you can disrupt this maternal behavior. So we can disrupt the maternal behavior of rats by not providing enough bedding for a nest. The rat mom will spend all of their time trying to find more bedding and rearranging that bedding because it's not working right. And she neglects to lick and groom her pups. And what we see is exactly what we've been talking about here, that you see those negative effects of emotional reactivity, stress reactivity. We might not say emotional in rats. Stress reactivity. You put them in a stressful environment, their cortisol goes way up. They're more distressed when they're separated from the maternal rat, that they can't regulate as well on their own, and that their brains are altered. And so it's all of that social interaction is clearly affecting brain development as well. That is where I wanted to stop, and I think we can take some questions, but I think all of us are going to come to a panel of questions here as well. So thank you for your attention.